Good morning. It's a pleasure to receive Dr. Robert Kaplan at our Conahp 2021. Today in Brazil is the Physicians' Day. We celebrate in the whole country. And today is a special day because physicians were doing a huge effort during the pandemics. And the pandemics make us do a virtual meeting. I thank Dr. Robert Kaplan for joining us. Dr. Kaplan is the Director of Research at the Clinical Excellence Research Center at the Stanford Medical School, and also had a huge contribution at the NIH as an Associate Director for Health Population. And not only this, but also a Chief Science Officer at the USA Agency of Healthcare Research and Quality. Dr. Kaplan, welcome to our meeting, and please go ahead with your special presentation.
Thank you. First of all, thank you for having me, and it's an honor to be able to speak to this group, particularly on Physicians' Day. So thank you. I'm going to talk to you today about sort of my views as a skeptic about a lot of what's going on in healthcare and healthcare delivery. And so I'm going to be going back and forth between some of the epidemiology and then what this means for hospitals and the way we structure and deliver healthcare. And so the bottom line is that I think that we need to have a much broader vision of what healthcare is attempting to achieve. So let me begin by saying that throughout the pandemic, we've been told to listen to the scientists. And so the argument is that we need to trust science, and science alone will lead us to the other end of this. But the difficulty is, as scientists and physicians, we don't necessarily agree with one another. In fact, data are conflicting, there are differences of opinion, and that the views that we held relatively recently may have evolved to something different. So in order to introduce this, I want to bring us back to two very famous British scientists, physicians. These two gentlemen, Dr. Archie Cochrane and Dr. Bradford Hill, Austin Bradford Hill, made the argument that we need to ask three questions about what we do in healthcare. The first question is, can what we do work? Or it's the can it work question. The second question is, does it work? And the third question is it worth it? And I'm going to be coming back to this frequently over the course of this presentation. So let's begin with the can it work question. So can it work is a binary question. The question is, does the treatment or what we're doing work or not work? And we might ask, well, what types of research are focused on this binary question, the can it work question? And again, it's sort of proof of concept. And my argument is that just about everything that we did at NIH gets at the can it work question. It's basic lab science, phase 1 human trials, phase 2 human trials, and phase 3 larger human trials. And in fact, the can it question has its roots in what we call health system 1.0. This is the kind of the original way we built the healthcare system. And here's what the healthcare system looked like. This is the healthcare system of yesterday. This is the first era. And the healthcare system was really focused on acute and infectious diseases. It was dominated by the germ theory of disease, and the time frame was very short. So the goal of medicine and healthcare was to deal with problems and deal with them relatively quickly. And the service that was offered was medical care. It was largely insurance-based, and the model was an industrial model. So you can see here that these hospitals, first of all, we had these wards with multiple patients in them, and hospitals actually looked somewhat like factories. And the goal was to reduce death. So let's just look at the germ theory. The germ theory of disease surfaced in the 17th century, and Louis Pasteur and others thought that there was a one-to-one relationship between microbes and microbe exposure and disease. And in fact, a lot of what we see, at least in the marketing for current therapies, is based on this understanding that if we just understand mechanisms, then that will lead to the remedies for almost all of these health conditions. So for example, there's a narrative that comes from Big Pharma, and you can see these two women here. The woman on the left is a lab scientist, and you can see here with her goggles. And then the woman on the right is a cancer patient. And the narrative is that there's this very smooth linear translation from bench to bedside, and that it happens very quickly. So we see advertisements. This one comes from the Toronto Globe and Mail. A couple of years ago, I went to the University of Toronto to give grand rounds, and that the marketing for their cancer therapy at the University of Toronto went something like this. They had a switchboard, so a panel with a lot of switches on it, and it said in their newspaper, if you want to get your cancer turned off, come to the University of Toronto and they'll flip the switch and you'll be okay. In fact, we see some of this currently. And so, for example, might ask the question, well, how well do we understand the relationship between vaccine-induced neutralizing antibodies and symptomatic COVID infections? And the reason that I raise this is that last week in the US, we had hearings on booster shots for the Moderna, Pfizer, and the Johnson & Johnson or Janssen vaccines. And that they were premised almost entirely on changes in neutralizing antibodies, as opposed to outcomes that are relevant to patients. And in fact, what came out at some of the hearings is that the mapping of antibody levels to infections, hospitalizations, and deaths is quite imperfect. So one of the issues that I wanted to raise for discussion was, well, how well is this very traditional model doing at helping us understand outcomes relevant to patients, that is, a symptomatic disease, hospitalizations, and deaths? And when I was at the NIH, I had the opportunity to serve on a couple different committees that were government-wide in the US that were looking at replicability in science and some of the challenges that we face with studies that don't replicate perfectly. One of the things that we came across was what's happening in a lot of bench science. And again, what we had come to appreciate was that that although we assume that bench science is very pure, in fact, there are problems. For example, one of the things that came out of that work was the realization that if you're a cell biologist and you're studying cells, it's very easy for those cell lines to get contaminated. So we looked at papers showing that guinea pig cells can masquerade as mouse cells, or that ovarian cancer cells are later found out to be breast cancer cells. This is because that within laboratory contamination is very common. And in fact, in recent years, it's been shown that cell line contamination occurs in somewhere between 14 and 30% of cell lines. And so cell biologists, when they purchase a cell line, are told that what you need to do is hire an authentication service to come out and document that the cells that you purchased are actually the ones that you think they are. Also true in a lot of preclinical research studies. So trials, for example, that evaluate therapeutic in animal models don't always replicate. So, for example, this slide comes from a very interesting organization which studies early phase studies of therapeutics for ALS. And what they did is they took published studies, which you see in the aqua bars, and then they replicated them. And the replications are shown in these black bars. And what they found is that none of the studies that were already published actually replicated. In fact, in most cases, the effects were very small, And in some cases, it actually went in the opposite direction. There's also a big problem with generalizability. And I really love this quote from Norbert Wiener that says, the best model for a cat is another cat, or preferably the same cat. Well, what could he have been talking about? Well, when I was at the NIH, I one time gave a talk and a seminar with Robin Barr. Robin was the director of extramural research at the National Institutes of Aging. And in his talk, Robin said, well, we've cured Alzheimer's disease 300 times in mouse models, but so far failed to cure it in any humans. And he was talking about a systematic review of 221 consecutive phase 1 placebo-controlled trials that failed to show benefits of therapeutics that had worked well in animal models, typically mouse models, but actually failed to advance in human trials. In fact, it's been argued recently that there are a lot of trials. For example, the recent therapeutic that was actually approved by the US FDA was something that worked well for amyloid in the brain, but actually was unable to show benefits in human trials with outcomes that were meaningful to patients. So the attrition rate for drugs based on clinicaltrials.gov in the Alzheimer's disease area are only about 3% or 3.4%. So what happened with this early phase? Well, we had a healthcare system that was built very much on the germ theory of disease. And we do know that germs are important. In fact, the current pandemic makes that very clear, that infectious diseases still are something that are of great importance to us. But in fact, most premature deaths are not associated with communicable diseases. And it's modified a little bit with the current pandemic. In fact, most premature deaths are associated with chronic diseases. And so what happened was that the way we thought about healthcare and healthcare delivery changed, and we had to modify the healthcare system to deal with that. So there were scientific changes, epidemiology in particular. We had the Framingham Health Study and other big studies, and we had to rebuild this healthcare system. And the healthcare system, sort of the second era, is the system that we have today. So the focus, although we still have communicable diseases, the focus is on chronic diseases. We have multiple risk factors, and so that's the foundation for a lot of what's done in healthcare. The time frame, as opposed to the original healthcare system, which was built on a very short time frame that is dealing with the infections, is now much longer. And that meant that the way that healthcare delivery had to change. It had to be prepaid benefits. The delivery model became much more corporate. The goal was to prolong disability-free life. And in fact, you can see on the left side that instead of hospitals looking like factories, in fact, a lot of the care moved in an ambulatory setting. So let's move on to the second question that Cochrane and Bradford Hill raised. And that's the question of, does it work? Well, we've just mentioned that there's been a problem of replication, but we also have problems with efficacy versus effectiveness. We know that there are exclusion criteria and inclusion criteria in trials that might not represent patients that you serve. And there are a variety of other problems that we need to address. So one issue is assumptions that we make about mechanism. And again, I think that the original model was based on the idea that people are like cars. In other words, if you have a problem with your car, you have a broken part, then you fix it. That seems obvious. And so the idea was, in the human body, if something's broken, then you replace it or you fix that part. But it doesn't take into consideration that cars are designed by humans and manufactured in factories. Humans are much more difficult to understand. So the assumption that fixing the broken part will actually remedy the problem gets at a variety of different issues. One of the issues that we've paid a lot of attention to is the surrogate marker problem. And again, you might say if the broken part is that your cholesterol is high or your blood pressure is high, the assumption is that if you remedy that problem, that is, you fix that one thing that's broken, blood pressure, cholesterol, or blood sugar, then in fact, the problem is resolved. But it doesn't take into consideration that the human body is very complex with many different feedback loops. So we know, for example, from large randomized clinical trials that fixing what appears to be off in terms of a biochemical indicator or a surrogate marker doesn't always fix the problem. So here, for example, we have the diabetes portion of the Accord trial, published really several years back. In this case, people with type 2 diabetes were randomly assigned to standard therapy or to intensive therapy. And you can see that if you look at the glycosylated hemoglobin, those in the standard therapy had higher levels than those in the intensive therapy. So in the intensive therapy condition we achieved the goal of fixing the broken part. We lowered glycosylated hemoglobin. So you might assume that this problem is fixed. But in fact, when you look at outcomes from the trial, just on the right side, then if you look at deaths from any cause, in fact, those in the intensive therapy group didn't do better. In fact, they did significantly worse. And there are just many, many examples. I have files full of examples like this. This is from the CREATE trial. This is a trial that looked at people who had low hemoglobin levels because of cancer or renal disease. And they are randomly assigned to take erythropoietin stimulating agents to increase their hemoglobin levels. And you can see group 1 who got the erythropoietin had an increase in comparison to those in the placebo group or the control group. And so we assume this problem is fixed. But the goal was to eliminate or delay the use of hemodialysis. And in fact, those who got the intensive therapy were more likely, rather than less likely, to end up on dialysis. In the cardiovascular area, one of the most challenging issues is intervention for people with atherosclerosis and coronary artery disease. So here you see this rationale. So the person has a lesion that you can see in the top. You insert a stent and open the stent, and that restores blood flow. And so the assumption is that angioplasty with stents fix the broken problem. So this problem is fixed. But this has been studied in a variety of large randomized clinical trials. And this one, the Courage trial, which is an older trial, was published in 2008, took about 2,300 roughly patients with objective evidence of myocardial ischemia and significant coronary disease, and it randomly assigned them to PCI or to optimal medical therapy. And the primary outcome was death from any cause or non-fatal myocardial infarction over an interval of about 2.5 to 7 years, an average of 4.6 years. And here are the results. So if we look at deaths from myocardial infarction or deaths from any cause, there was no benefit whatsoever of stenting. And this really caused a lot of concern among interventional cardiologists. There were a lot of methodological problems in the Courage trial, but there's a new trial which is out online. So it's published online in JAMA. It was in May and should be out in print version very soon. This is the ischemia trial. And in this trial that was done at 320 sites in 37 countries, over 5,000 participants at a cost of over $100 million. People with coronary disease, the symptomatic, were randomly assigned to either coronary artery bypass graft surgery or to stenting, and then they were followed prospectively. And here are the results in this new JAMA paper, that if you look at cardiovascular disease deaths, there's 13.3% in the stent or CABG condition and 15% in ordinary medical care. This is not statistically significant. And similarly, deaths from any cause, 6.5% versus 6.4%. And this was very much, although it's completely consistent with the Courage trial and several other trials, this was actually very much a concern to interventional cardiologists who had tremendous confidence that PCI and coronary artery bypass surgery would have profound effects. So one of the assumptions that we make from basic science is that most good ideas will eventually become good and successful pharmaceutical products. And in fact, this is sort of the rationale for a lot of what we did at NIH, is turning discovery into health. Over the last couple of years, I've been writing more opinion pieces mainly for the popular press that try to get at what's going on, what assumptions we're making, and how we have to revise these assumptions to come to more realistic, have more realistic views about medicine and healthcare. So one of the issues is that we make the assumption that if it's a good idea in the laboratory, it's likely to become a successful pharmaceutical product. And in fact, that people in the US feel that the regulatory agencies like our Food and Drug Administration are just getting in the way, that it's slowing down the time it takes to get a successful product into the hands of practicing physicians and into the bodies of patients. So one of the things that I had observed when I was at NIH, Francis Collins had a blog, and his first blog that he offered looked at what we call the therapeutic pipeline. And in this, we take compounds that have therapeutic promise. And what Francis mentioned in his blog was that if you take 10,000 compounds, and this is in the drug discovery phase, all of these are patented, and all of these appear to have great potential. Of those 10,000, only about 250 of them, this is 250 of the 10,000, make it to the preclinical phase. So most of these promising ideas actually are abandoned because of various problems early on. And then that's only about 2.5%. It's about what you would expect from a one-tailed t-test. And then if you take those 250 compounds that get tested in preclinical studies, only about 2% of them, a little bit less than you would expect by chance, make it into large-scale clinical trials. And then after that, only one of the 10,000 compounds actually makes it to the point where it's a licensed pharmaceutical product. So now let's take those that actually become licensed and look at what happens to them post-marketing. I had the opportunity when I was working at the NIH to look at large clinical trials. And we looked at every large clinical trial that the National Heart, Lung, and Blood Institute at the US had funded over the last 50 years. And what we noticed was that most of these trials, by the way, dealt with products that were licensed or at least very near licensure. And what we discovered was that most of these trials showed that the therapeutic agent worked. So if the bar or the box is black, that suggests that that was a positive trial. And if it's gray, that was a null or negative trial. So most things that were tested in those big trials actually did work. But then something happened. And this is, by the way, I'm sorry, this is before the year 2000, where this graph comes from. Then in the year 1997, the US FDA passed the FDA Modernization Act. And in that act, they created transparent reporting standards. They initiated a service called clinicaltrials.gov, which I know you use also in Brazil, where trials have to be listed prospectively. And they required all grantees to register their trials starting in the year 2000. And so we looked at the likelihood that trials would produce a positive result prior to the year 2000 or after 2000. And here's what happened. After the year 2000, the likelihood that trials that the National Heart, Lung, and Blood Institute sponsored had a significant result actually declined, and declined rather dramatically. So I'd like to just take a couple minutes and talk about some of the issues that these are methodologic issues that remain a challenge for us. And one is the issue of generalizability. And so we know, for example, that people who participate in randomized clinical trials are different from the people that are cared for in clinical practice. So this slide comes from an evaluation that was done by investigators at Stanford, and they were looking at trials in alcohol, tobacco, and illicit drug use. And what they showed was that that common exclusions in these trials were problems like psychological or psychiatric disorders, medical compliance problems, age, social instability, distance from the clinic, education, and so forth. And in fact, these are problems that you would expect to be of high probability for people that are have difficulty with alcohol, tobacco, or illicit drug use. And what this analysis showed was that between 64 and 96% of potential participants were excluded from these trials. This one is a systematic review of participants in studies on Alzheimer's disease. And what you see is the age ranges, that is the mean and 95% confidence intervals for the subject populations in these studies. And and this roughly is what the age distributions look like. But here we have the average age of people with Alzheimer's disease. And what this shows is that people who participated in the studies are all systematically, sometimes by decades, younger than those who actually have Alzheimer's disease. This one, a similar idea, this is the SPRINT study. The SPRINT study was the largest study that I think has ever been done on the treatment of people with low spectrum blood pressure. So SPRINT was the one that suggested that it's valuable to push systolic blood pressure below 120 millimeters of mercury. And it's had a profound effect on guidelines in the US and in other countries. But in fact, the eligibility criteria for SPRINT systematically eliminated about 95% of the people that might have been eligible for this treatment. So there have been concerns like this that have been raised in the most recent hearings on COVID boosters. So in the US, for example, the studies, there were 3 hearings before the FDA in the last few weeks. That one of the big issues is the original studies only followed people for about 2 months in the Pfizer and the Moderna trials. And so the generalization that these vaccines were 95% effective, by the way, I do think the vaccines are wonderful products, but in fact, they do seem to lose efficacy over the course of time. And we wouldn't have known that because the trials actually ended their follow-up in at least the trial phase after 2 months. And in the booster studies, they ended after 1 month. And in the Israeli boosters trials, it was only 12 days. The data that were presented in the last few weeks had surprisingly small sample sizes. In one component of the Pfizer booster study, there were only 11 people between ages of 16 and 64, and only 12 over 65, so a total of 23 in that phase of the study. And in the phase 2, the part that was used for the emergency use authorization, it was a total of 306 people. In the Moderna study, it was 344 people. And these results are generalized to, as we see on the right, literally hundreds of millions of people. There's also a concern about data manipulation. And we make the assumptions that scientists are objective, dispassionate, completely honest, and so forth. But in fact, we know there are problems with reporting results selectively. One of my favorite examples comes from Germany, where they had looked at, it was an agency that recommends funding for different services. This was an evaluation of serotonin, selective serotonin reuptake inhibitor, and people were randomly assigned in a variety of clinical trials to take the medication or to take placebo. And that the company that produced this medication presented data on 2 trials. And these are the 2 trials. Both of them showed that there was a significant benefit of the medication. But this German agency raised the question about, well, what happened to the other trials? So we know, and they knew that a variety of other trials had been conducted, and they said that they weren't willing to recommend this drug unless they got the data from all trials. And they did get the data. And what you see in the other lines are the other trials. In fact, an enormous amount of the data was hidden from public view. There's also the concern about spin. And we know, for example, that there are a variety of serious biases. One is there's citation and reporting biases. So in this example, this is also in the depression area. This is a group of studies identified through clinicaltrials.gov that evaluated treatments for depression. And the green dots represent positive trials, and the red dots represent trials that were null or negative. And it was about 50-50. About half the trials showed that the treatment worked, and about half showed that it did not. But then if you look at what studies get published, you can see that almost all of the positive studies got published, but only about half of the null or negative studies got published. And then there's outcome reporting, which I'll get to in a minute. And spin means that the authors interpreted the results as more positive than they actually were. Let me just show you this issue with spin. These are cardiovascular trials. And they look at the prevalence of spin in published cardiovascular trials, where they know that the result was statistically non-significant for the primary outcome. And these come from parallel group randomized clinical trials in the 6 journals with the highest impact factors. In all of the trials, the primary outcomes were non-significant, but in reporting, the authors actually were able to find a positive spin. In other words, although the results were null or negative, then it appeared that the authors reported something more positive. In fact, in 67% of the cases, the authors make a statement suggesting that the treatment does work in the main part of the study, and 57% in the abstract. And actually, even 10% of them, there's a suggestion that the treatment works in the title. So I'm going to move on. In fact, I'm going to try to wrap up in a few minutes so we have time for questions. But I just wanted to get at the issue of, is it worth it? So again, Bradford, Hill, and Cochrane said, does it work? Can it work? Does it work? And is it worth it? So we have to ask this question from a patient's perspective. If it's approved by the FDA or another regulatory agency in your country, patients do want to know, will it harm them, how much benefit they can expect, and how much will it cost? And I'll just give you one example, sort of my favorite one. This is a medication for a condition that's called female hypoactive desire disorder. This is essentially a loss in sexual interest among women. And that they evaluated this treatment based on a self-rated sexual desire scale that went from 0 to 6.0. And the data they presented to the FDA showed that on this scale that goes from 0 to 6, it changed about 0.2 on a scale. So a tiny, tiny change. But there's side effects. In fact, if you look at the percentage of women experiencing nausea, it was over 22% among those taking the medication, as opposed to 3.9% among women taking placebo. And further, this is not a pill. In fact, it has to be injected. And so, and it has to be injected 45 minutes prior to sexual activity at a cost of about $899 per month. So the advertisement for this was give her the power to call the shots, actually quite literally. I'm maybe running a few minutes behind. I can skip through this next phase if or maybe I'll just try to do it quickly, just to say that I think that we all need to develop greater skepticism in the way that we evaluate clinical data. This, for example, shows the relationship between LDL cholesterol levels achieved through use of statin medications and clinical events on the y-axis. And so you might ask the question, well, how strong does that relationship look? Well, I think to most of us, not very strong. But if you look at this, and then if you look at it in relation to what's been shown in advertising, in this case for PCSK9 inhibitors, it looks like there is a very strong relationship. And in fact, this slide has been used to make the argument that in fact, it's necessary to push everybody's LDL cholesterol down below about 70 milligrams per deciliter. And that you hear this repeatedly, that this is a linear relationship. But it turns out that this linear relationship, first of all, is not based on individual data points. It's based on treatment and control groups in selected clinical trials. And in fact, this line that you see actually comes from these same data that I just showed. Here, though, the regression lines are in for primary prevention and for secondary prevention trials. So what is clear is that in secondary prevention trials, people who have vascular disease or have had a previous myocardial infarction, lipid lowering works very well. But in fact, when we look at this, again, this is the same data, it turns out. They actually decided to ignore the primary prevention trials, which is a big slice of them. And in fact, when we look at populations that should be or shouldn't be given statins, the data are much more confusing than you might think. So for example, if you look at all-cause mortality in one big meta-analysis of 11 randomized clinical trials, and there's another meta-analysis that will be published soon that's very similar to this, you can see that in fact that there is not a significant benefit for extending life expectancy in these trials for overall mortality. And there are just a variety of different examples like this. One of the big issues is, should statins be used for older people older than 70 years old? And it's been suggested that they should, because if you look at the ASCVD risk calculators, basically everybody over 70, or at least men over 70, all of them come into the risk category, and virtually all of them end up on statin medications. But if you look at the trials for people over 70, in fact, the benefits are not statistically significant. And so, again, I won't go belabor this, it's the same result. So there does not appear to be a significant benefit for mortality for older patients. Now, one of the big problems is that in these trials, there aren't very many older patients. In fact, the major trials that are included in the meta-analyses include very, very few, and in many cases, no people over the age of 70. So I'd like to just turn to my last few comments, and this has to do with this theme that we had, the more than medicine. I wrote a book a few years ago that tried to get at, well, what do we need to do with the healthcare system? We had 1.0 that dealt with primarily germs, 2.0 that's our current healthcare system. Where do we need to go from here? And the argument is that the future is arriving, so that the focus now is, although we still have to deal with communicable diseases, we still have to deal with chronic illnesses. But in fact, what we need to attend to is broader than that, because what determines life expectancy is much more than just risk factors and communicable diseases. And so we know, for example, that the very best predictors of how long people live reside outside the healthcare system. Here's just one example of a study that I worked on with George Howard, who's at the University of Alabama in Birmingham. We had a data set of about 30,000 people in a study on stroke that we followed prospectively for about 30 years. And we looked at the very best predictors of how long people live. And it turns out the very best one is how many years of education people have. So that if you look at that in the US, having a college or graduate degree in relation to people with less than a high school education, it turns out that the difference in life expectancy is about 12 years. And I'll come back to putting that into perspective. And you can adjust for demographic factors, income, risk factors, and behavioral factors. All of those attenuate this relationship, but none of them get rid of it. We know that the deaths of despair in the US, these are drug overdoses, suicides, and so forth, are best predicted by years of education and not by traditional medical variables. And then just to put this into perspective, if you look at how much life expectancy you gain from having a pap smear every year over every third year, you actually can't even see that benefit. If you look at a mammography versus no mammography, a tiny benefit, there is one. Cholesterol, a couple months. Blood pressure, a little bit over a year. Diabetes management, a year and a half. But whether or not you're employed versus not employed, it's about 6 years. Smoking is about 7 to 10 years. Having a high school education, less than a high school education versus graduate degree, it's a dozen years. And living in a high income area versus a low income area in the US, this is in Philadelphia, it's actually 20 years. I mean, it's just an enormous difference. I'll skip this just by saying we've analyzed social determinants of health in a variety of ways. They all come out showing that social determinants of health have a huge impact where healthcare has a relatively small but important impact, about 10% versus over 40 to 50% of the variance in outcome. So I just want to close by mentioning that as we change to go to this new system, we actually need to change the way we think about things and the data we use and so forth. I just wanted to bring up this example of how poor our data are in helping understand this. And this example is from the biennial censuses in the United States. So in the United States, every 10 years, we count the number of people in our population. And we count them by race and so forth. And it determines the number of congressional seats and resource allocation and so forth. And these censuses started in 1790. And the question is, well, how has the demography of our population in the US changed over the course of time? Well, it turns out we really don't know, because the race was not self-reported until 1960. And so until 1960, census takers just looked at people and they decided what race they were. And then if we look at what happens over the course of time, so categories, I'll just move this. Categories come in and out of this. And so, for example, that the category Mexican was added to the census in 1930, but it was taken out again in 1940. Korean Hindu was removed in 1950. And then 1960 self-reporting was allowed. And then Asian and Pacific Islander groups were put together in the same category in 1980. In the year 2000, people were allowed to select more than one race if they wanted to, and that turns out to be a huge portion of the US population. About 10 million Americans changed what race they were in when they were allowed to self-report. And we know that genetic ancestry changes people's view of their own race by about 15 to 40% in some cases. The category Negro was removed not until 2020 in the US. So all of this suggests that, in fact, we don't know very much about the demography of our population, because in every census since the beginning of the census in the US, in fact, how we classified race has been different. So let me just wrap this up by saying that we have these three different ways of looking at outcomes. It's the can it work question. These are laboratory studies, surrogate endpoints. The does it work question. These are pragmatic trials, real world conditions. And then the is it worth it question, which requires a completely different way of looking at things. Policy studies, cost utility studies, shared decision-making in clinical practice, and so forth. And so as we move forward, the way our hospitals and healthcare settings are structured, the way we deliver care changes and needs to continue changing. We need to adapt to the realization that although we need to confront germs and risk factors, in fact, we have social determinants which have a huge impact on the way we structure and deliver the services. So I'm going to stop. I went a little bit over the time I had anticipated, but thank you once again on Physicians Day for having me, and I'd be happy to answer any questions.
Okay, thank you, Dr. Kaplan, for the presentation. It is very interesting. I wish we could have more time for many questions. I will address only 2 questions because of the time. That's the main reason. And how can we correlate the COVID event that changed, brought a great urgency in the healthcare discoveries to bring solutions? So how do you assess the impact of the pandemics on how we develop the healthcare system and the efficiency of delivering healthcare?
Yes, thanks. It's a wonderful question, and I think it's going to have a big impact. So let me just mention 2 areas that I think are important. One is that what we've learned in the US is that remote clinical medicine works really well. And, it doesn't work well for everything, but patients can get tests in the lab and they can interact like we're interacting on Zoom. And it actually seems to be desirable for clinicians and desirable for patients when it doesn't require hands-on face-to-face encounters. It doesn't work for everything. And I have a feeling that is here to stay, and that's going to affect the way that healthcare looks, how many exam rooms you need and so forth. And I think that that's going to evolve. The second area is that I do think that there have been concerns about, how do we get from a vaccine theory to a delivered vaccine in a short period of time, which, it's quite impressive how that was accomplished, and very good products got to the market quickly. On the other hand, as I pointed out, most products don't work. And, we're starting to see with therapeutics now, it's going to be very challenging to get things to market, but still assure efficacy and safety, because we can't do the trials that quickly. And that, in fact, through the history of medicine, there are, unfortunately, numerous examples of things that caused harm, and that we do need to find ways, perhaps using real world evidence trials and other things, to get evidence, to make decisions quickly, but not to relax methodologic standards.
I see and agree. But on the other hand, do you think the acceleration mode will be more used regarding chronic disease or disease that bring a population impact? You said you brought many examples on vascular disease, and we would bring as much as we can in cancer. And the acceleration mode would be helpful for those who need an answer and don't have time to wait. We experienced this on COVID, but many examples in the medical field are available. Do you think we will use more agile models in the future?
First of all, thanks for the question. It's a very challenging question. And unfortunately, I'm not going to, let me just say, I'm not going to give you a very clear answer. It's a very clear question. I don't know that I have a clear answer. And here's why. In the US, we have accelerated approvals for cancer therapeutics. And that following the 21st Century Cures Act, which was passed at the end of the Obama administration, we have an accelerated pathway for cancer therapeutics. And that there is an increasing number of products that are now licensed on the basis of a single randomized clinical trial. And sometimes they're small clinical trials. what's starting to emerge is that some of those products are being recalled. And so they've had to back off some of those approvals. So there have been some unanticipated harms that have come out. The actual evidence base for some of those approvals is very weak in relation to traditional approvals. And so we definitely need this balance between desire to get products to market quickly, but then the realization that regulatory agencies do have this serious responsibility to protect the public. And that over the course of decades, that pendulum has swung back and forth, and I think that we probably in the US were overly rigid between the 1970s and 1990s because of fear of serious harm. And then I think now it's probably swung in the other direction, in that in my opinion, we've approved some products that produce more harm than benefit. And so this is a debate, and not everybody agrees with it. And I'm sure that that's going to be a hearty debate for the years to come.
Okay. Well, we need to end here. I wish we had more time to bring interesting questions from the organization and also from the audience, but we can't. Time is up. So I would thank you for a great presentation and sharing your experience in managing a healthcare system. Thanks a lot, Dr. Kaplan.
Yes, and thanks so much for having me. It's a real honor to be able to do this, particularly on Physicians' Day in Brazil. So thank you.
Thank you.
Goodbye.
Bye.